Hello everyone, welcome to the class. Today we're gonna do laboratory 3 about the determination of blood glucose. So I just want to ask you first, can you differentiate between blood plasma and blood serum? So today we're gonna use the biological sample called serum. So first you should know what the difference between the plasma and serum. The way of glucose determinations, you can use Benedict test as you have learned already, I think maybe in your high school. But today we're going to use the enzymatic assay to determine the blood glucose concentration. Why we need to use the enzymatic assay to do this laboratory? So this is the question, and you should discuss this in the report. So the laboratory today we have two experiments. The experiment 1 we're gonna prepare the standard curve of the glucose concentration. Before the experiment 1, I'm gonna show you the equipment and the consumables that we're gonna use today. So we have the auto pipette. So you may have learned from Ajahn Tong already, and you have the tips for used with the pipette. You have auto pipette with different size, and you got the UV, not UV, this is the visible spectrophotometer here. You got the vortex here for mix the solution together. You got test tube. You got the biological sample, and you should stay the solution on the ice box, because the biological sample, if you leave them at the room temperature, all the content in the biological sample can be degraded. And what we got for the experiment 1 is the standard glucose concentration with different concentration, start from 0 milligram per deciliter, 100, 150, 200, and 400. So you got 5 concentration of the standard glucose concentrations. For the first step from the lab manual, we're gonna switch on the spectrophotometer first. So the first step, you need to check whether the equipment is on or not. So you should switch on first to warm the lamp before you use the spectrophotometer. And then from the protocol, you're gonna prepare the 5 test tubes. So these are the 5 test tubes that you're gonna prepare. Then you label using the permanent pen. Don't label into the white part of the tube here. So you should label out of the white area, because if you label into the white area, it's gonna be like this, and it's difficult to wash it out. So I'm gonna label 5 tubes. Tube 1, 2, 3, 4, and 5. Okay, you got 5 tubes already. So now you're gonna add the reagent called glucose oxidase reagent. In the reagent is contained the enzyme that will convert glucose to the product, and this product can be absorbed the light at the absorbance of 500 nanometer. So this is the glucose oxidase reagent. So we're gonna pipette 1 ml and 500 microliter. So I adjust the volume of auto pipette to 750 microliter. It means that you just do 2 times when pipetting, then you get the final total volume of 1000 and 500. So I'm gonna use my finger to hold the cap like this. Don't place on the table. It's gonna have the contamination with the other thing on the table. So I hold the cap like this. So I pipette. And then place the solution into the tube. So this is for tube 1, and we do this for 5 tubes. So we already have the 5 test tube prepared, and we already add the glucose oxidase reagent already, 1.5 ml. So the next step, we're gonna add the standard glucose concentration. We use the smaller autopipette to pipette 10 microliter of each standard glucose concentration. The first one is 0 concentration, so we use distilled water basically. When you add the solution, you add this solution directly into the reagent buffer like this. Not add into the side wall of the test tube because the volume is so tiny, so you should add them directly into glucose oxidase solution. Change the tip when you're gonna pipette the other concentration of the standard glucose. So the next one is 100 milligram per deciliter. And add them straight away into the second tube. If you are struggle with the side of the pipette, so you can just bend the tube a bit and then add them directly into the glucose oxidase reagent. The tube 1 is for the concentration 150. Sorry, tube 3, not tube 1. And tube number 4 is for the concentration 200. And the last tube is for the concentration of 400. Now we got to mix all the solution by vortexing. When vortex touch the top part of the tube, not touch like this, otherwise it's not have any force, so you touch the top part. So now you already done preparations of the solution for the measurement with the visible spectrophotometer. You're gonna leave this solution for 10 minutes at room temperature or ambient temperature. So now we got to transfer all of the solution after the incubation at 10 minutes into the plastic cuvette. This is the size of 1.5 ml. So we're gonna pour all the solution into the cuvette. So this is the blank solution. Before you're gonna insert your cuvette into the spectrophotometer, you need to wipe around the cuvette. In the case of contamination of something, when you touch or something like this, it's gonna hide the light that passes through the cuvette. And before inserting the cuvette into the spectrophotometer, you need to check the path light. So the path light is in this vertical direction. So you need to turn around this side to match with the light part. So this is blank solution. So you're gonna set blank by place zero absorption. So now, if you read the value here, you should read the value of 0. So this is already done. So the second one is tube 2 that you already have 100 mg per deciliter. So you just transfer the solution by pour them directly into the cuvette again. Then wipe out around the cuvette. Put the cuvette in the spectrophotometer. Then you can now read the OD value directly from the machine. Then you do like this for the third, the fourth, and the fifth tube. Then you can get all of the OD value from the 5 tube here in this table. Okay, from the manual in the experiment 2, we're gonna test the concentration of 5 different samples. So the first one, you prepare 6 test tubes like this, and then you pipette 1.5 milliliter of the glucose oxidase solution into 6 test tubes. I have already prepared all 6 test tubes with the glucose oxidase reagent already. So for the tube 1, we're gonna add blank, which is distilled water for 10 microliter. So I use pipette to pipette 10 microliter of the distilled water into tube 1. And this is the blank sample. The second tube is normal control. So for the sample, I have already kept the sample in the ice box to avoid the degradation of the content in the biological sample. So the second one is the normal control. So you pipette again 10 microliter. And add into tube 2. Then for tube 3, you pipette the abnormal control. The tube number 4 will be the unknown sample 1. The tube number 5 is the unknown sample 2. And the last one, you can shoot the standard glucose concentration, just one concentration, to add into tube 6. So I shoot 200 microgram per deciliter to add into tube 6. Now you have finished adding all the sample solution 10 microliter into the 6 tubes here. So you just mix them together by vortexing. Okay, after we mix the solution together, so we leave the solution for 10 minutes already. So now you can see the color change in each tube that contain the glucose concentration. So now we're going to use the spectrophotometer to measure the concentration of glucose in each tube, in each sample, I mean. So for the blank solution, you couldn't see any color change in here in tube 1. So for the spectrophotometer, we have already set the maximum wavelength for the measurement at 500 nanometer. So you pour the solution directly into the cuvette again. Wipe around the rim of the cuvette, then measure. So this is the blank solution. So you press 0 absorbance again. So now you should have the OD value of 0 for this solution. The next one, the second tube, is the normal control. So you pour the solution directly again into the cuvette. Wipe out around the rim and then measure. So for this tube, you can read the OD value directly from the spectrophotometer. Then you do this step for the rest of the test tube. And then you can get the OD value. And you should repeat again, start from the step 1 that you prepare another set of 6 test tubes. And then you add the glucose oxidase reagent. You add the 6 samples like this, and then you mix them, leave them for 10 minutes and measure again like this. So you can get the OD value from the 2 or 3 sets of the experiment, and then I will show you the result in here in the table. So for now, we come to the end of the laboratory 3. So I just want you to answer the questions for this lab in the report. And in the report, the first, you should plot the standard curve using Excel software. And the question is, what is different between blood plasma and blood serum? And the second is, do the sample come from the diabetic patient? Which one? The unknown one or unknown 2. And 3, discuss the principle of the assay in brief, not 10 pages like that, just 1 page, no more than 1 page. And the second, why we need to use this enzymatic assay instead of chemical assay like Benedict's test or Philip reagent. Thank you very much for your attention.